This session today is sponsored by DuPont, and the title will be The State of Disinfection Byproducts and Health. And our presenter is Manolis Kogevinas. He graduated from the Medical School of Athens, Greece, and did his PhD in epidemiology at the University of London. He worked at the International Agency for Research on Cancer, and he is co-director of the Center for Research in Environmental Epidemiology in Barcelona. He's a joint professor. He has a joint position as professor at the National School of Public Health in Athens, Greece. And his research focuses on evaluation of environmental, occupational, and genetic factors in relation to cancer. Please welcome Manolis.
Thank you, Terry, for the introduction. And very glad being here. It's the first time I come to this conference, and we have been working in my group for quite a few years on disinfection byproducts in drinking water and lately also in pool water. And I will give an overview of what we know, well, my interpretation of what we know on disinfection byproducts and health. And I will talk indiscriminately between drinking water and pool water, knowing that is not exactly the same, actually. So just to start, some basic things that, as I mentioned also just a few minutes ago in a discussion we had, are not, actually I don't have to prove in this audience about these things that chlorination is a major advance in public health for the last 100 or something years. And also physical activity, including physical activity that we're doing in pools, is of major public health importance. And we have to keep this in mind when we are talking about the potential adverse health effects of being exposed to disinfection byproducts and health. So, and in the end, we have, I think, to have in mind that we have to have clean drinking water and pool water, and at the same time, I think the message is, because of the potential health effects, we have to try to minimize the chemical exposures. So, and that's what is my message when I'm talking to the media, when we are doing studies on pools or whatever else. So I will go through several things. So I will have to be rather brief on each of these items, on cancer, on birth outcomes, asthma and allergies, and I will present the results of the swimming pool, the experimental study we did in Barcelona on measures of joint toxicity and respiratory effects, and some issues on future research. So DBPs, we identified them a few years ago. There are hundreds of them, and we, at least in the EU, we regulate the 4 trihalomethanes, and this is the level we accept in drinking water. One of the problems we have in our research is that we have been measuring the 4 THMs, and these are the ones up there. So, but these are the compounds that Susan Richardson identified from the pool water, which is just underneath my office. And if you do any complete analysis of pool water or drinking water, you identify something similar, hundreds. So one of the problems we have in research is that especially for research on cancer that goes back many tens of years, is that we have not been able to evaluate all of these, and we evaluate these ones. It may be okay, because they may be a marker of the whole pool of disinfection byproducts, but it may not be also okay. They are not necessarily a good marker always. And also, the distinction between chloroform and the brominated is not standard in different parts of the world. So we have this problem, and we have been working also with Michael Playwell, who will talk after me, about how we could evaluate the toxicity of the whole mixture. So this is a pending issue. Also, we have been using methods, experimental methods, like the Ames test. This is, again, from Susan Richardson paper, and this is from the pool underneath my office. And if you do any analysis of a chlorinated pool or a brominated pool, well, you will find that the water is mutagenic. If you do any analysis of drinking water that's chlorinated, you will find that it's mutagenic. So we know that there is a potential for toxicity. The fact that it's mutagenic, similar to the air, if you go out, get a measurement of the air outside the hotel, It is mutagenic. The fact that something is mutagenic, that there's a marker of toxicity, is not the same as saying there is disease. This is a marker. And we're using it in research. It's an interesting marker because it is informative. We're using it in research. The fact that we find these markers of toxicity cannot be immediately interpreted in measurable clinical outcome, health outcomes. And we have sometimes problems in communicating this. So we are exposed to disinfection byproducts by drinking the water, and this is more important, for example, for alloacetic acids. And we are also exposed by dermal absorption and inhalation. And these are very important routes for swimming pools. We get massive exposure to trihalomethanes, to the more volatile compounds, inhaling them, and also from dermal absorption because we're doing physical activity. Most of the studies we had for many years on cancer was basically on ingestion. And we were very late in incorporating in the epidemiological studies these other routes of exposure that are very important. So let me go to the issue of DBPs in cancer. And basically the evidence is for bladder cancer. This is the analysis that Cristina Villanueva did, who was doing her PhD. Now she's an assistant professor at my center some years ago, summarizing all the evidence we had at that time on bladder cancer. And that was from metrics of exposure that looked only on ingestion. How much water did you drink? Was it chlorinated? How much chlorinated was it, etc. And you can see, it doesn't matter what type of statistical model it is, but you can see an increase in risk with more THMs. And we have pretty high levels in some of the areas where the studies have been done. These are studies in the US and in Europe. So after that, we published the results from the Spanish bladder cancer study, which was the first one to incorporate inhalation, ingestion, and dermal absorption. We asked for how frequently did you shower, how frequently did you bathe, et cetera, et cetera. And it's the first study actually to evaluate these routes of exposure that for many of these compounds are the most important. And we replicated, again, actually, what was found from the previous studies, and also for inhalation and ingestion. And at that study, we also were the first ones to have this question of swimming pools. And when we published the results, we were very aware that we had not done a very good work on evaluating exposure to swimming pools. It was the first time it was done. We had a couple of questions. It's not easy to do this, looking back 30 years. So we were very cautious in interpreting the results. But we had this result. So we had a 60% increase in men and women who went to swimming pools. So that was preoccupying. But we were very cautious because, the methods we used, the questionnaire we used was not so elaborate. But for this reason, we actually started the more detailed studies, more sort of experimental studies on swimming pools. And we're still waiting for the study that the National Cancer Institute is doing in New England to see whether they will replicate these findings. The other thing we have now, we have the possibility of using a lot of the new biotechnology, genetics. So if this is, for example, the population response to an exposure, this is risk, we can try to differentiate people who may be more susceptible or less susceptible to the effect of an exposure. So the less susceptible would respond, to same amount, but they would need to have a higher dose to get the same amount of risk. And say the more susceptible would just get the effect with a lower dose. Can we distinguish between us concerning DBPs? Well, there are some actually genes, some enzymes that metabolize the DBPs. So we looked at that in the bladder cancer study that we're doing it with the National Cancer Institute. And we based our evaluation on experimental studies that have already been done with, in the laboratory using Salmonella. And they showed that genetically modified Salmonellas would have a different response depending on whether they expressed or they did not express this gene. It doesn't matter what it is, a glutathione transferase, so T1. So depending on whether they were genetically modified or not, they would have a differential response to the chemicals. And we know that this happens for other chemicals. So we did it in the bladder cancer study, and this is what we find. This was published by Ken Cantor a couple of years ago. And we found an increased risk, 20%, 100%, 120%, in those people of the population who were expressing the gene as compared to those who were not expressing it. You can see here, the reference is 1. This is say, if you're above 1, you're at the risk. If you're below 1, you're not at the risk. So in this group, we don't find much. And in this group, we do find an increase. So the increase we had found in the overall study was mostly confined in a subgroup. It's a big subgroup, actually. It's about perhaps 30 to 40% of the population. So this is interesting, and this gives more credit, actually, to the findings that probably what we found is true. The other cancer where there has been a lot of research is colorectal cancer, and it's a very important cancer, a very common cancer. And here the results are not very consistent, actually, as I interpret them. There may be different interpretations. We have some experimental data. Much of it has been produced here in the States of preneoplastic lesions in the colon in animals. So you do find preneoplastic lesions depending on you expose them to bromate or to brominated compounds. This is not cancer, it's preneoplastic, but it's interesting because colon is a disease where you have preneoplastic lesions and then you go to neoplastic lesions. So it's an interesting model. So what do we find in the epidemiological studies in humans? This was a meta-analysis. Well, you put all the results together and you try to get a combined estimate done a few years ago. And these are the cohort studies, and these are the case control studies. Cohorts are prospective, case controls are retrospective. And the cohort studies, they find, well, nothing significant. It's not, we have measures of the error, so it's not statistically significant. And for the overall, these authors concluded there's a 27% increased risk for colorectal cancer. Now, many of these studies, the quality of the evaluation was not very good, actually. So many of us, maybe it's correct what they're saying. Maybe there is a 30% increased risk. But for many of these studies, actually, the evaluation of the exposure, it's complex. You have to go back 30, 40 years. It's not easy to do. So for many of these studies, the evaluation of the exposure was not, to my opinion, done properly. So I'm really waiting for some new studies that have more advanced models of exposure before I personally conclude. We are doing one of these studies. We're not the only ones doing these studies. We're doing one of these studies. It's a new study in Spain, and we're trying to do very, very extensive exposure modeling, which is crucial here. And we'll see what will come out. We haven't got the results yet. And I hope there will be other studies that have done extensive exposure modeling because it's crucial. So for DBPs, for bladder cancer, the evidence we have is pretty consistent. For levels that are above drinking water above 50 micrograms per liter, you have a clear increased risk. For colon, there are several positive studies, but the evidence is weaker. And for other cancers, we have few studies or very inconsistent evidence. I don't think we can conclude anything. So let me move to birth outcomes. There are many studies on birth outcomes, many. So studies that have looked at fertility, and I'm using slides that Sylvain Cordier used at the Gordon conference we had a couple of months ago. Sylvain Cordier works at INSERM. It's like the French NIH, and she's among the very good researchers on birth outcomes and DBPs. So that's why I have her logo here. So there are some studies on male fertility. We have practically nothing on female fertility. We have some studies on fetal loss and stillbirth, some studies, quite a few studies on birth defects, and many studies on fetal growth, things like low birth weight, small for gestational age, this type of outcomes. And overall, the findings are really not consistent, or they are negative. But maybe we don't have good enough studies. So I'm not going to go into details because I don't have enough time. I'll just show this biggest meta-analysis, so synthesis of all the studies that James Grelier from the Imperial College did, together with Mark Neuenhuizen, putting together the findings of all the studies. So this is the one. If you are on the right, you have an increased risk. If you are on the left, you have a decreased risk. So these are the studies we have, and the error bars are, the random error of the estimates. And this is the summary. So you see it's very, very close to 1. It is actually 1.01. And it's because we have lots of studies, it is statistically significant. So which means you have, for 10 micrograms per liter of THMs, you may have an increase in risk of 1%. This is the conclusion of this meta-analysis, and it's statistically significant. The issue, so basically, it is so close to 1 that it's very difficult to interpret. You cannot say there's not a statistically significant increase. There is. There's a 1% increase. The issue is, and that's how James actually interpreted the findings. And when he wrote the paper, and is this of clinical interest, such a small increase in risk or not? Maybe yes, maybe not. But it's a very, very small increase.
So what is the standard in Europe?
Well, this is... Well, in Europe, the standard is 100 micrograms per liter. Yeah, it is higher than the US. So this is expressed increase in risk per 10 micrograms per liter. So if you have 10 micrograms more on average in your pregnancy, during the 9 months of pregnancy, you will have a 1% increased risk of having a smaller baby than what it should have been. So the issue is here, can we interpret this small risk? I think it's an important finding. The clinical relevance is this, we can discuss about it. We have now the results of the European study. Unfortunately, they're not published, so I cannot show them here. And this will be among the most actually strong studies we have, because we have 13,000 mother-child pairs with very extensive exposure evaluation. And now we will incorporate some of the information that Michael Plewa has done, and he will show what he's doing, so you will understand that it will be much more informative. One of the issues that have been discussed, though, concerning this, for example, this 1% increase that I was saying about, is that, okay, it's 1%, it's not clinically relevant, but we have to take into account that the information on exposure may not have been good enough. If the information on the exposure is not good enough, the risk could have been even higher. It could have been lower, but it would have been even higher. So Sylvain Cordier and some other people say, why don't you use a biomarker rather than trying to reconstruct the exposure? The problem is that we don't have good biomarkers. We don't have good biomarkers. We can measure them in the environment. We don't have good biomarkers for humans. We can measure them. We can measure THMs in the blood. We can measure them in the urine. We can measure them in exhaled air. But, exhaled air, well, you go to a swimming pool, if you don't measure them immediately, it's gone. So we don't have long-term biomarkers like we have for dioxins, for example. Dioxins, you go 20 years later, you measure it. So one of the biomarkers we have refers to alloacetic acid, and it's the TCA. And there are some studies that have shown there's quite a good correspondence between TCA concentration in tap water and TCA concentration in urine. You can see the correlation is not great, but it's there. And we have 2 studies, one from France and one from China, that have looked at biomarkers, and they both actually have found a positive effect, an increase of about 80% of fetal growth restriction with TCA measured in urine, and again, a decrease in birth weight in China. So, is the fact that James, when he did his meta-analysis, didn't find practically anything because the 1% is not interpretably clinical, is it because there's nothing, or is it because the exposure was not measured correctly? So we'll have to get some better studies, actually, to respond to this question. And also, I just want to give this study because it's interesting for the wider discussion we have of risk and benefits. This is a study based on the Danish national birth cohort, huge cohort. 100,000 mother-child pairs, huge cohort. The Danes, for those, because most of you are from the US, they register everything. So it's easy to do these very complex studies, big studies. This is not a small thing to have 100,000 mother-child pairs starting 3rd month of pregnancy and following the children, until adolescence or more. So, and in this study, they're doing a lot of bicycling in Copenhagen. Those of you who have visited Copenhagen, you will see people going biking, and they go a lot to the pools. So they looked at the pregnant mothers, whether they went to the pools, whether during the pregnancy, whether they did bicycling, or whether they didn't do physical activity. And these are the results, and they're interesting, because they don't have much DBPs, actually, in Denmark. They chlorinate very, very little. Although in the swimming pools, we don't know the levels, but they should have, but they haven't analyzed them. But what is interesting to see is that the risks are all for the active ones, whether it's swimming or bicycling. Actually, you have a lower risk of preterms, you have a lower risk of small for gestational age, you have, well, nothing much for birth weight, and you have a lower risk of congenital malformations compared to those who do not exercise. And that's interesting. We have the same effects in adults. You exercise, you have lots of positive findings. So when I'm referring to that we have to look at the costs and benefits, I'm also referring, to findings like that. If you are a public health agency, you have to take into account specific health risks. You have to have the wider picture. You have to minimize the specific health risk, bladder cancer, but you have to have the wider picture. You have to promote physical activity. This is what is coming out also from this study. So results are basically inconsistent for most of the outcomes. That we have some indications for stillbirth, for some birth defects, and for fetal growth, what I showed before. Some outcomes are not studied. Female fertility has not been studied, and it should be studied. And basically, most of the studies have focused on THMs. And I think we have to do studies that will focus on other exposures. So let me move to asthma and allergies. Very, very controversial issue, and very difficult to evaluate. It's difficult to evaluate. So we have case reports on occupational and accidental exposures. We know that people in swimming pools, guards in swimming pools, whatever, they may develop asthma through different mechanisms, not necessarily through an allergic mechanism, through very high exposure to irritants. We know that. We also know that elite swimmers have more airway inflammation, they have more asthma, and similar to people who do cross-country skiing, they that also have, similar to other sports that do endurance sports, and so they do have more asthma. No, but the question for the general population is, how does this translate into the general population of adults and children? And for the children's studies, that's where is the controversy. And actually, I don't agree with some of the criticism that are being done to some of the studies that are produced in Belgium that minimize the effect of the Belgian studies. Although I'm critical of some of these studies, for those of you who have followed the literature. So this is a meta-analysis, again, a synthesis of all the studies done a few years ago. Now it's out of date. It's 2008. We have many studies after that. I mean, this meta-analysis concluded, putting everything together, that we had a combined risk of 0.82. 0.82 compared to 1, so 18% down. So you don't have a risk, you have a prevention, actually, for children who go to pools concerning asthma. And then they went and excluded the Belgian studies by the group of Albert Bernard, and I personally don't agree with this type of way of dealing with the data. But they did it because mostly the positive studies mostly come from Belgium. But there are not necessarily evident biases in this study, so that's why I don't agree with excluding. But in any case, putting everything together, you have a decreased risk. And you have in some studies increased, but most studies have a decreased risk. Just to put an example from some of the Belgian studies, this is total time in a pool attendance, so less than 50 hours, 50, 100, 120, more than 200 hours. And this is, again, your prevalence of asthma depending on whether you are atopic, depending on levels of your IgE or not. And, there's basically an increase, whatever if you're atopic or not, of your asthma prevalence. And these are standard findings. I'm just putting one slide from the findings, but they have several of these studies. And we, in Spain, in Barcelona, we haven't found it, and other people haven't found the same thing. But, we still have these positive studies. So we have to, to be careful how we interpret the data. All these studies are actually what we call cross-sectional. You get the children, you see whether they're asthmatic, whether they have eczema, whether they have other allergic diseases, and then you ask the mothers about their swimming pool attendance. And the problem of some of these studies is that your behavior concerning what you do as an exercise may depend on your health status. So you may be an asthmatic child and you may not go to pools, or you may go more to pools because this is a recommended exercise. And if you do it retrospectively, it may be difficult to disentangle what's first and what's after. So this is the criticism, but still, we have some studies cross-sectional that are positive and some that are negative. And, my studies are negative, but the fact that my studies are negative doesn't mean that I'm correct. The Belgian studies are positive. And there are some other positive studies around. And there are other negative studies. Now, this is actually, I think, the most informative study we have. And this was done by one of our PhD students in collaboration with a big child cohort in England, in Bristol, the ALSPAC cohort. It's a great cohort. They have followed children from birth. Now they're 16 years old. And there are about 16,000 children. We had information for swimming pool for about 5,000, 6,000 children, and it's prospective. So we had the information on swimming first, and then children were followed up so we could see what happened afterwards concerning disease. So in this study, which is the strongest evidence we have, actually is negative. We find no association with asthma, and in fact, we find an improvement of the FEV1, of the lung function of the asthmatic children if they swim. So it is not great still, even in ALSPAC, the questionnaire is not great. It's the best we have, but it's not great. So, I think it's the best evidence we have, but, there are question marks here. And then we should have more studies like this. But this study actually made the recent evaluation by the COMEAP. This is the advisory committee to the UK government on air pollution that includes swimming pools. It was actually one of the pieces of the evidence that they used to say that although we have experimental evidence showing that asthma can be produced in the swimming pool, so there are the mechanisms there, the epidemiological evidence is weaker now than before. That's what this committee said. And part of it was because of this study. So where are we, altogether? Now, most, but not all, epidemiological studies find that swimming in pool is not associated with asthma in children. There is some evidence on eczema, and there is also some evidence, which is very interesting, I think, on using biomarkers on effects on the lung epithelium with Clara cells. CC16 is a protein, and it's interesting because it talks about mechanisms. However, most studies are cross-sectional, and it would have been really preferable to have prospective studies. And I think this is high priority for research. There are lots of children going to pools. We have some positive findings. We have more negative than positive, but we have some positive findings. We have to take them seriously because there are lots of children going to pool. And we have to get some really big, very good study here. So let me move on the biomarker-based study. This is, I think, again, one of the best experimental studies we have. We have to get, again, more of these studies. We did it in a swimming pool in Barcelona, in 2 swimming pools in Barcelona, and we looked at markers of genotoxicity. We don't look at disease. We look at markers of disease. Markers of genotoxicity and markers of respiratory effects. This was published a couple of years ago and was quite a lot in the media. So this is a design. We have 1 week without swimming for those of the volunteers who were swimmers, 40-minute swim, and then we got samples, or we look at some lung function before and afterwards. And then 2 weeks later, we take another sample of urine. So we look at exhale air, so we can measure THMs in the air. We get blood, urine, we get exhaled breath condensate, so we can get from the deep lung, fluid from the deep lung. And we look at exhale NO, which is a marker of eosinophilic asthma, and we do lung function. And this is a swimming pool, so it's actually a very good public pool. It's big, it has big windows, and these were the volunteers, 50 volunteers, and we measured in water what you see, THMs and chloramines, in air THMs and trichloramine, and exhale air THMs, and you can see how we were measuring exhale air. And the biomarkers of genotoxic effects are biomarkers, and I'll show one of them, the micronucleus. We do COMET assay, micronucleus in blood and in urine, and also urine mutagenicity. So they are well-used biomarkers. We know about them. We have been used many years. This is a micronucleus. So when a cell is dividing, sometimes we have errors. And so part of the nucleus stays outside the nucleus. And this could be a whole chromosome, or it could be part of a chromosome. So it is an error when the cell is dividing. And we know from adult studies that it's not such a marker that you can use at the personal level. It doesn't make sense to do your own analysis to see if you're an increased cancer risk. But at the population level, we know from big studies in Europe that those populations that have more of macronuclei, they have 10, 20 years later more cancer risk. So it is a good marker to use. And that's what we found. Basically, we found increased macronuclei after swimming the pool, basically for the brominated compounds. And we were surprised because, it is a not trivial thing to say that you go 40 minutes in a pool and then you get an increase in your macronuclei. And we also find increased urine mutagenicity, again for bromoform, basically. So these were the findings, and they have been discussed a lot, and they have to be replicated. And now we have actually got funding to replicate with much more elaborate techniques. This was the first study. There were lots of things to do better in each study. All the studies have errors. So, but this is what we find. So this is, interesting findings, actually. Not totally surprising that we find it. I was surprised that we found it after, 40-minute swim. Actually, before the study, I thought we would find the inflammatory responses. That would be the thing we would find, rather than the genotoxic stuff. And it went the other way around. So, we have a strong association of brominated compounds with swimming and markers of genotoxicity in blood and urine. And, we really need to replicate. And it would be better if some other group replicated it. Maybe they are replicating it. But we definitely, with the European project, we are trying to replicate these findings with new subjects. So respiratory and inflammatory effects, this has been discussed. So we did a number of biomarkers, the CC16, that looks at lung permeability. And a number of cytokines, inflammatory markers, and we looked at this in blood and in exhaled breath condensate. And basically, we have some findings similar to what the Belgians are finding on CC16, and all the rest is negative, which was surprising to me. So these are the two positive findings of the study. So we have an effect of an increase of CC16 protein with exhaled breath, one of the brominated compounds. You can see the association. And obviously, we also have an increase with energy expenditure. What happened in this 40 minutes? What did the people do? They were exposed to DBPs, and they did physical activity. That's what happened in these 40 minutes. And we know that with physical activity, you may do lots of things. Genes get expressed, and others don't get expressed, et cetera, et cetera. So we had measured physical activity. We knew how many laps the person did. So it was an indirect measurement, but it's good enough. And we have an independent effect of both energy expenditure and of one, at least, of the THMs in relation to C16. And this is the only positive finding. All the rest was entirely flat. Everything else concerning the inflammatory markers, which was a surprise to me. And so no association of exposure during swimming and markers of inflammation or oxidative stress. I didn't talk about oxidative stress, but there's no association. And a positive association with CYS16, which is what the Belgians are finding also. So what are the future research I think we should be doing? We have to have studies, both experimental and the long-term epidemiological studies, cohort or case control. We have to do a better job with exposure assessment. It's crucial. Maybe it's okay to measure only THMs, but we have to see what else we could measure to characterize better toxicity. And I think the swimming pool study will have to be replicated. We always replicate studies if you want to conclude. And I think, and there's an American epidemiologist, Kyle Steenland from Emory, who is would like to do a study. I don't know if he has got the funding. One should look at long-term swimmers. What is the long-term cancer risk of people who have swum, many years of their life? If what we find in the population studies is true, we should find it actually in people who have swum for many years. It is not as, it may be a bit more complex because professional swimmers or people who swim a lot will be of better health than people who don't do physical activity. Whatever the chemicals in the pools, people who do, if they are. Yeah, sure. The latency is longer, but people who do physical activity are better off than people who do not do physical activity. So you cannot compare people who swim a lot with people who do nothing. It doesn't make sense. I mean, they will come out well. What you want to see is whether people who swim as compared to people who do another physical exercise, people who do bicycling or something, have more bladder cancer risk. This is the question. So, and it would be really nice if we could get, also, I don't know if the association can help in this and bring together registers that exist and do a long-term follow-up. What happens with people who have gone a lot to the pools? That would be a very nice study. And obviously, for the respiratory thing, we have to get prospective studies. These are not easy studies on the fact that we have diverging results. It can be because of differences in methods and because it's not easy. We're not talking many times for heart outcomes. Wheezing. Wheezing is not an easy symptom to evaluate in children. It's really not easy. I have done lots of these studies, and you can get a lot of differences of how people report wheezing. So you have to have very good protocols. If you can do that prospectively, so much the better. And we had a recent report, I don't think it's published yet, on endocrine disruption among people going in the swimming pool. It has not been published actually yet, but it will be published. So that would be another interesting outcome to keep in mind. This is the water research group. So Cristina Villanueva, who should have been here actually doing this presentation, she's 8.5 months pregnant. So I'm here instead of Cristina. Well, I'm directing the whole center. Actually, one of the directors of the whole center, but Cristina is directing the water research group. And this is our center. It's in the middle of Barcelona in front of the beach, a very nice location. So if you come to Barcelona, come and see us there. Thanks a lot.
Thank you, Dr. Kogevinas. We'll have a short question session here. Please keep them short and concise because I have to condense them to repeat them for everybody. Any questions? Over there.
In your one slide there where you talked about that study that was done in Britain and the asthma was not caused by swimming. Did you study the chlorine level at all?
No.
The question was about the study that was done in England and whether chlorine levels were examined, chloramine levels.
No, it's a big study, and it's a questionnaire-based study. The good thing was that it's prospective. So they asked the mothers every year questions. Not all the questions every year, but we asked first, are your child going to swimming pool? For how long? And then we followed up the children. So we're looking whether the children who, at 3 years old, they said they were going, and those that were not going, what happened to the children in the future? It's like an experiment in a way. But we don't have levels. For swimming pools, for population-based studies, it's more difficult because you have lots of swimming pools and you have to have, how can you do it? So that's, in a way, you can get these very more precise exposure estimates in smaller populations, experimental studies probably, but in these big studies, you may try to get some estimates, but they will not be very accurate.
What if they start at 6 months?
What?
What if they start at 6 months?
The question is, what if they start looking at these people at 6 months of age?
I mean, this is a real question. Is baby swimming good for health? Or should the kids start, should we start putting the kids in the pool after 6 months? The Belgian committee, they thought, they were very harsh on the studies of Bernard, actually, the committee by the Belgian government. And the evaluation of the Belgian committee was that, there's no clear indication that the babies gain by going, before 6 months to the pool, in fact. So they learn to swim, well, yeah, they can learn it after the 6 months. So this is what the Belgian committee said.
Just wondering, had you also, someone looked at UV sanitizers and UV, and then any effect on being exposed to, I guess, once you run water through there, you'll have a lot more free radicals?
Yeah.
The question is, when they were doing these studies, did they look at whether the pools that the people were using had UV systems on them?
No, or ozone or whatever. No, they haven't been done. I think we should have studies, also experimental studies for pools using different disinfection methods. This is an obvious thing also to do. Although basically we are chlorinating nearly everywhere, but still there are alternative methods, and we should be evaluating these because if in some of them, in fact, we find that we don't have effects, this is an important message to give. But we don't have the studies.
No more questions? Okay. Oh.
Ask about standards, like the ones that do relate to trihalomethanes. I heard that Denmark, for example, has got 20 micrograms.
The question is about the various standards that have limits on trihalomethanes. Are they a factor?
Well, many countries in Europe actually have fairly clean water to start with in the rivers, so they chlorinate the drinking water very little, or they just don't chlorinate. You have non-chlorinated water in some parts of Europe. So they have very, very low levels. In Spain, we have among the worst, in the coast, the Mediterranean coast of Spain, we have them among the worst waters in Europe, actually. So we have strong findings in Spain, maybe also because we have big exposure contrast, and to find differences in risk in a population, you also have the people having exposure contrast. If everybody's exposed to the same, you don't have anything. You may have effects, but you don't find them. So we have big exposure contrast in Spain, and we find these effects. In Australia, from where you come from, you are the champions in THM levels. You have the highest THM levels in the world.
Yeah.
So, but in Denmark and these places, they have very low levels because they don't need to they don't have precursors in the water.
I've got 93% of Australian competitive swimmers use Ventolin. That's the definition of asthma. 93% is a huge number.
Any other questions? All right, thank you for attending. Thank you, Dr. Martin.